For part 2, let's talk about ovogenesis. Ovogenesis takes place in the ovary. Ovary, it's here. Here the first ovary and the second ovary. And begin during fetal life, continue after puberty, and is completed only its fertilization occurs. Ovogenesis can be divided into three main phases: multiplication, growth, and maturation. The first phase is multiplication. During fetal life, the primordial germ cell differentiated into ogoni. This ogoni divide by mitosis, increasing the number. At this stage, each ogonium is diploid, contain 46 chromosomes, which is mean 2n. The purpose of this phase is simply to produce a large number of ogoni. The second phase is the growth. The ogoni increase in size and become primary oocyte, also called oocyte 1. Each primary oocyte is surrounded by a single layer of flattened follicular cells, forming primordial follicle. The primary oocyte still contain 46 chromosomes, which is mean 2n, and then enter meiosis 1, but the division stops in prophase 1. This arrest lasts from fetal life until puberty. For phase 3, maturation, which begins at puberty. Every month, under hormonal stimulation, one primary oocyte continue meiosis 1, and at the end of the first mitotic division, the primary oocyte produce secondary oocyte, oocyte 2, and first polar body. Both cells are haploid, contain 23 chromosomes, which is mean, but each chromosome still consists of two sister chromatids. For secondary oocyte, which is mean oocyte 2, immediately begin meiosis 2. However, it stops at metaphase 2. This is the cell that is released during ovulation. If the fertilization does not occur, the secondary oocyte is die. If the fertilization and the sperm enter the secondary oocyte, which is mean the fertilization take place, the entry of the sperm triggers the completion of meiosis 2. The secondary oocyte then divide into ovatid and the second or the secondary polar body. The mature ovum contain 23 chromosomes, which is mean N, with one chromatid per chromosome. For formation of zygote, the nucleus of the ovum fuses with the nucleus of the sperm. Okay, the nucleus of the ovum fuses with the nucleus of the sperm cell. Since each gamete contribute 23 chromosomes, the resulting is zygote contain 46 chromosomes, which is mean 2n. This zygote is the first cell of a new individual and will begin dividing by mitosis. So, for conclusion, ovogenesis begins with an ovonium containing 46 chromosomes through mitosis, growth, and two mitotic division. Only one functional ovum is produced, and the first mitotic division form the secondary oocyte and the first polar body, while the second mitotic division is completed only after fertilization, producing the mature ovum and the second polar body. Finally, the fusion of the ovum and the sperm form a diploid zygote with 46 chromosomes. Before we continue, it's important to understand the difference between ovogenesis and folliculogenesis. For ovogenesis is the process of forming the female gametes or eggs cell. Folliculogenesis is the process of growth and maturation of the ovarian follicle that surrounds and nourishes the developing oocyte. In the other words, the oocyte develops through ovogenesis, while the follicle develops through folliculogenesis. These two processes occurs together inside the ovary. Let's begin with the first phase, called primordial follicle. Is the smallest and most immature follicle found in the ovary. Inside it, there is primary oocyte surrounded by a single layer of flattened follicular cell. The primary oocyte contains 46 chromosomes, which is mean 2n, and is arrested in prophase 1, okay, of meiosis from fetal life until puberty. This means the oocyte remain inactive for many years. It is stimulated by hormone. Next, the primary follicle, which is mean the primordial follicle, develop into a primary follicle. The follicular cell become cupidal and begin to multiply. Okay, a protective layer called zona pellucida start to form. This is zona pellucida start to form around the primary oocyte. The oocyte still primary oocyte and still contain 46 chromosomes and remain arrested in prophase 1. The next stage is secondary follicle. The follicular cell continue to proliferate, so it's continue to proliferate, forming several layers known as granulosa cell. This is granulosa cells. Around the follicle, two connective tissue layer appear. Theca interna, it's here, and theca externa. For theca interna, which later produce steroid hormone, and theca externa, which provide structural support. The oocyte is still a primary oocyte with 46 chromosomes, and it remains arrested in prophase 1. For cavitary follicle, as the follicle continue to grow, small fluid-filled space appear, it's here, and merge to form a large cavity called the antrum. This stage is known as the cavitary follicle or enteral follicle. The follicle becomes much larger and the oocyte remain surrounded by zona pellucida and granulosa cell. At this point, the primary oocyte is still arrested in prophase 1. Follicular phase. Now we enter the follicular phase of the ovarian cycle under the influence of hormone FSH. One follicle becomes dominant and develop into the mature Graafian follicle. Just before ovulation, the primary oocyte completes meiosis 1. It produces one secondary oocyte and one first polar body. The secondary oocyte contains 23 chromosomes, which is mean N, and immediately begin meiosis 2, where it stops at metaphase 2. Around day 40 of a normal 28-day cycle, the level of LH rises sharply, which is mean, or this event is called the LH surge. The LH surge causes ovulation. During ovulation, the mature Graafian follicle rupture, releasing the secondary oocyte. This is secondary oocyte. This secondary oocyte is then captured by the fimbria at its opening of the uterine tube and enter the fallopian tube where fertilization may occur. Remember, the cell released during ovulation is the secondary oocyte, not the mature ovum. After ovulation, The empty graphene, it's here, the empty graphene does not disappear. Instead, it's transformed into a temporary endocrine gland called the corpus luteum. Okay, this is corpus luteum. The corpus luteum secretes large amount of progesterone and a smaller amount of estrogen. Okay, this hormone prepare the endometrium for possible implantation of a fertilized egg. About 7 days after ovulation, the corpus luteum reaches maximum size and activity. If fertilization does not occur, it gradually degenerated over the following day. By the end of the menstrual cycle, it becomes a small fibrous scar called the corpus albicans. As progesterone and estrogen level decrease, the uterine lining breaks down, result in menstruation and a new ovarian cycle begins. Now let's talk about spermatogenesis, which is the process of sperm production. It takes place inside the testis, here, tubule inside testis. For step 1, spermatogonium, which is a diploid cell 2N, it divides by mitosis to produce more spermatogonia. Some cells remain as stem cell while others developed into primary spermatocyte. For step 2, primary spermatocyte, the primary spermatocyte is also diploid 2N. It undergoes meiosis 1 to produce 2 secondary spermatocytes, each contain 23 chromosomes. For step 3, each secondary spermatocyte undergoes meiosis 2. This produces 2 spermatid, so 1 primary spermatocyte form 4 spermatid. Okay, here 4 spermatid. For step 4, the spermatid do not divide anymore. Instead, they change their shape in the process called spermiogenesis. They develop an acrosome, a head, a midpiece rich in mitochondria, and a tail. Finally, they become mature spermatozoa. Okay, and thank you for watching.